Hello, this class is to serve as a review for the exam on the endocrine system, endocrine anatomy, physiology, and endocrinology. That is happening next week. Next Tuesday, let me check my calendar, April 28th is the day of exam 3. And this class is to serve as a review for the exam. This slide shows one of the key concepts that we have been using over and over again in this section, the idea of positive feed forward and negative feedback. So this is a generic slide showing that from the hypothalamus, hormones are released into the median eminence, into the portal circulation of the median eminence, and that blood flow from the infundibulum, from the median eminence of the hypothalamus to the anterior pituitary, triggers the release of other hormones from the specific cells of the anterior pituitary. And in many cases, those hormones circulate through the systemic blood to other glands to trigger the release of other hormones from those glands that then have an effect on cells in the body. There are many exceptions to this general positive feed forward. For example, the somatotrophs of the anterior pituitary, the somatotrophs, release growth hormone, and growth hormone has effects by itself. It does not require the activation of other glands, does not require the activation of other glands to have its effect. The liver releases IGF-1, which is a cofactor for growth hormone, but growth hormone does not trigger the release of IGF-1 from the liver. But the standard pathway is, let's say, GnRH, gonadotropin-releasing hormone, is released from the hypothalamus. It triggers the gonadotropes of the anterior pituitary to release LH and FSH. LH and FSH cause the cells of the gonads to, well, in some cases, some of those cells respond to LH to synthesize androgens, and other cells of the gonads respond to FSH to proliferate or to help the production of other cells, proliferation of the granulosa cells in the ovary, or the stimulation of the Sertoli cells in the testes to aid in the production of sperm. And then androgens and estrogens are produced by the cells of the gonads, and they have effects on the gonads themselves as well as throughout the body. So that's a more, typical pathway, but growth hormone follows this pathway. These hormones have negative feedback. The hormones produced, say, by the gonads have negative feedback on the pituitary and on the hypothalamus, and in some cases that I haven't articulated clearly, the pituitary hormones have negative feedback on the hypothalamus. But there are exceptions to negative feedback, of course, because, for example, in female reproductive endocrinology, at ovulation, the negative feedback of estrogen on the production of GnRH and the negative feedback of estrogen on the production of LH and FSH is converted into positive feedback and becomes a positive feedback loop that leads to the surge in LH and FSH associated, or that causes ovulation. So bottom line, this slide is an important conceptual slide to understand, and it would be important to understand when this slide is accurate and when there are exceptions to the rules suggested by this slide. If we were together in class, I would start with a couple of clicker questions just to get the ball rolling and to get the question and answer stimulus running. So let's go ahead and do that. Let's do a few clickers. Which of the following hormones are released at the median eminence? So this is an anatomy question, asking you to know the names of some of the hypothalamic hormones that cause release, that are released in the median eminence and travel through the portal circulation to the anterior pituitary. So I'll give you a few seconds to see if you can answer this. The answer is GHRH and somatostatin are released by the hypothalamus into the portal circulation. And both of these hormones have effects on the somatotrophs of the anterior pituitary. Growth hormone releasing hormone, GHRH, stimulates the release of growth hormone from the anterior pituitary, and somatostatin inhibits the somatotrophs of the anterior pituitary and inhibits them from releasing growth hormone. So this is a slide that illustrates both the anatomy and some of the knowing the names of the cells, knowing which hormones are released where, and what they do is an important thing to know for the exam. All right, the next question. Which of the following hormones do not follow a feed-forward pathway? That is to say, the feed-forward pathway, the standard pathway is hypothalamus to pituitary to some other gland. Which of these is an exception to that feed-forward rule? The answer is insulin and parathyroid hormone. The stimulus for the release of insulin from pancreatic beta cells is high blood glucose. It's not a hormone from the anterior pituitary. The stimulus for release of parathyroid hormone from the parathyroid glands is high serum calcium levels. It's not a hormone from the anterior pituitary. So those are two, and there is at least one more, glucagon, and maybe there are others that I'm not thinking of right now, that do not follow the feed-forward pathway. Cortisol and thyroid hormone both do follow the hypothalamic pituitary, another gland pathway, because the stimulus for cortisol release is ACTH from the corticotrophs of the anterior pituitary, and it receives a stimulus from the hypothalamus, CRH. So CRH from the hypothalamus stimulates the corticotrophs of the anterior pituitary to release ACTH, which then triggers the cortex of the adrenal gland to release cortisol. So that's a classic feed-forward pathway. And the same sort of thing for thyroid hormone. TRH from the hypothalamus stimulates TSH from the anterior pituitary that stimulates the release of thyroid hormones, T3 and T4, from the thyroid gland. So those are the classic feed-forward pathways. So in class, I would ask if you have any questions, and I still am asking if you have any questions. If you have questions, please use the forum on Moodle or email me your questions, and I will be happy to try to answer them. In some cases, your questions might go off onto a detail or a tangent or a clinical setting that I don't know about, but I'll try to direct you towards sources to learn about them. In some cases, if you email me a question and I think it's on topic for everybody, I will post it to the forum. So please ask your questions. The other thing that I do in class, and we'll do now in this distance learning sort of thing, is to go through a case study. So let's read this together. Natasha Schick is a 23-year-old aspiring model who has always dieted to keep her weight in an acceptable range, quote unquote acceptable. However, in the past 3 months, she has lost 20 pounds despite a voracious appetite. She complains of nervousness, sleeplessness, heart palpitations, and irregular menstrual periods. She notes that she is always hot. I think that she feels warm, and she wants the thermostat set lower than her apartment mates. On physical examination, Natasha was restless, noticeable tremor in her hands. She's 5'8" tall, but she weighs only 110 pounds, which I think even for a model is considered low. Her arterial blood pressure was 160/85, which is high. Her heart rate was 110 beats per minute, which presumably is at rest, which is also high. She has a wide-eyed stare. And the terminology for that would be exophthalmia. And her lower neck appeared full. These characteristics were not present in photographs taken a year ago. So based on her symptoms, her physician suspected that Natasha had thyrotoxicosis, increased circulating levels of thyroid hormone. She is hyperthyroid. However, it is unclear from the available information why her thyroid levels were elevated. Laboratory tests were performed to determine the etiology, the basis of her condition. Okay, so here are the laboratory results. T4, total T4 was increased. Free T4, that is to say that the T4 hormone that is not bound to a carrier protein. So total includes both free and bound. Free T4 was also increased. And TSH, thyroid stimulating hormone, was low, so low that it was undetectable. So the physician considered the following possible causes of thyrotoxicosis. So, and based on his understanding of the hypothalamic anterior pituitary thyroid axis, he considered a hypothalamic tumor that was releasing too much TRH. He considered a pituitary tumor of the thyrotrophs of the anterior pituitary that was releasing too much TSH. He considered primary hyperactivity of the thyroid gland. And he considered whether the patient was taking exogenous thyroid hormone, which has been used but is definitely not recommended for weight loss. So using the laboratory findings and your knowledge, which one, let's just say, which one do you think is most likely? Let me stop here. I'm going to stop the recording so that you can pause and try to think about which of these is likely or unlikely and provide a suggested diagnosis. So which of these causes is most likely? The answer that I think is correct is C, primary hyperthyroidism, which might be Graves' disease. So let's see why A, B, and D are unlikely. If there was a hypothalamic tumor, producing too much TRH, you would expect there to be high levels of TSH. and that's not what is seen. It's difficult to measure TRH, but not impossible, and so the physician might have asked for a TRH measurement in order to rule that out completely. But a high TRH would normally be followed by a high TSH, and that's not what is seen. So, and that also rules out B. If there was a pituitary tumor producing too much TSH, well, then the low TSH would not make sense. Primary hyperactivity of the thyroid gland is likely because does that explain these lab results? Yes, increased T4, and decreased TSH because of excess negative feedback. Ingestion of exogenous thyroid hormone can't be ruled out, but the patient is concerned about weight loss, and it seems, and the physician presumably asked the patient, are you taking anything that has thyroid hormone in it? And hopefully the patient was honest with her physician. So the best answer is C, primary hyperthyroidism. So let's go through the follow-up. Well, let's think, before we go through the follow-up, let's think about some of the symptoms. High thyroid hormones high levels of T3 and T4 would explain the nervousness, the rapid heart rate, the fact that she feels hot because it increases cellular metabolism, and so she feels hot because of hyperthyroidism. It does explain the nervousness of the heart rate, blood pressure, which follows heart rate. The exophthalmia is a prominent sign of hyperthyroidism, and the fullness of her neck could suggest early stages of a goiter. Her thyroid is hyperactive, the cells are proliferating, and her thyroid gland is making her neck look swollen. All right, so let's go through the follow-up. Natasha had a thyroidectomy. Her hyperactive thyroid was removed, and it was successful. So she is recovering. Her nervousness and palpitations disappear. She is gaining weight. Her blood pressure returned to normal. Presumably, she is now taking thyroid hormone. You can take pills of thyroid hormone that, because she's no longer producing thyroid hormone anymore, her thyroid glands have been removed, she still needs thyroid hormone, and she's taking pills as a supplement, or really a replacement for her own thyroid. So she's taking thyroid hormone pills. She begins to experience new symptoms, alarming new symptoms: muscle cramps, tingling in her fingers and toes, numbness around her mouth. So she returns to her endocrinologist, and who finds that her total blood calcium concentrations, 7.8 milligrams per deciliter, that's total. Ionized, which means unbound calcium concentration, was 3.8 milligrams per deciliter. So 3.8 milligrams per deciliter of free ionized calcium and 7.8 milligrams per deciliter of total bound and free calcium. Both of those are lower than normal. Normal values are around 9 milligrams per deciliter bound or total and 5 milligrams per deciliter of free. So she has hypocalcemia. What is causing it and what should be done? So think about what happens when you remove the thyroid glands and why this might be causing hypocalcemia. I'll just stop talking here. I will just be quiet so that you can think of the answer, and then we'll go to the next slide. if we were doing this in class, I would say turn to your neighbor and try to figure this out, do a pairwise, work it out together. And then I'll call on some pair to provide their answer to the question. You can't do it in pairs, although if you want to, you can pause the slide and uh, email or text your friends and see if you can do it together, but we'll just move on. She, in the process of removing her thyroid gland, her parathyroid glands are removed. They're embedded in the thyroid gland, and so removal of the thyroid gland also removes the parathyroid glands, and you'll recall that parathyroid is important for maintaining a set point of ionized calcium in the blood. So you'll recall this antagonistic control system where if serum calcium goes low, then parathyroid hormone levels are high. Low serum calcium stimulates the parathyroid hormone glands to release parathyroid hormone, and it inhibits the parathyroid gland from releasing calcitonin. So the combination of high parathyroid hormone and low calcitonin causes increased calcium reabsorption from the kidney, increased calcium absorption from the intestine, increased calcium mobilization from bone, and so ionized plasma calcium concentration increases. And when it gets back to normal, then the concentration of calcitonin and parathyroid hormone they aren't necessarily the same, of course, but they are having the same effect to maintain a specific level of ionized calcium. But in this patient, the parathyroid glands have been removed, so the effect of parathyroid hormone is attenuated, and we now have a new set point, which is too low. And so the treatment of this would be to give parathyroid hormone supplement as well as thyroid hormone supplement to return to normal parathyroid hormone levels and a more normal calcium set point. So this problem was, the first part of the problem was for you to think about thyroid hormones and the feed forward as well as feedback controls. And the second part of the problem was for you to think about these antagonistic controls that lead to cellular or whole body homeostasis. So these were examples, and my recommendation is to understand these examples and maybe even think about applying these examples in other endocrine settings. Let's move on to the next slide. More clickers. Which of the following drug types could be effective in treating insulin-sensitive diabetes mellitus? So this is a form of diabetes where there is too low release of insulin. This is type 1 diabetes. The pancreatic beta cells have been destroyed. It's usually autoimmune, and there is too low insulin release. So which of these could be effective? Let me just say that in most cases of diabetes mellitus, not all of the beta cells are destroyed, so there are some. So think about the remaining few pancreatic beta cells. Which of these it would be effective to help those cells release insulin? I'm just going to be quiet for a minute, let you answer the question. So would a drug that inhibits activation of calcium channels assist the release of insulin? I hope you know that voltage-gated calcium channels are needed for the release of insulin, so that can't be a good answer. A drug that inhibits the GLUT2 transporter. You might recall that the GLUT2 transporter is the uniporter that brings glucose into the pancreatic beta cells. And so a drug that inhibits the GLUT2 transporter is going to inhibit the signal for the initial signal that triggers the cells to release insulin. So that can't be a good answer. A drug that inhibits leak sodium channels. Okay, so leak sodium channels are maintain the, assist in the maintenance of a resting membrane potential. Resting potassium channels and leak sodium channels combine to maintain a resting membrane potential. If there is inhibition of the leak, then there is less sodium influx. So that would be less of a factor for depolarization. So that would cause hyperpolarization. Hyperpolarization is not going to assist in the release of insulin. So that's not a correct answer. Okay, let's see if there's any correct answers. A drug that inhibits the ATP blocked potassium channel. Okay, if the drug inhibits a K channel, a potassium channel, it inhibits the potassium efflux, which is important for hyperpolarization or polarization. So inhibiting a potassium channel, including the ATP blocked potassium channel, would cause depolarization, and depolarization could be helpful in assisting the remaining pancreatic beta cells to release insulin. So my guess is that B is the correct answer. And yes, B is a correct answer here. A drug that inhibits ATP blocked potassium channels. So that now we have two ways to inhibit those K channels. ATP, when glucose is absorbed, and the drug. So it's an assist in the blocking of those K channels. And that would assist in the depolarization, which would assist in the activation of voltage-gated calcium channels, which would assist in the release of insulin, the secretion, exocytosis of insulin from pancreatic beta cells. Which of the following drugs could enhance glucose uptake? Okay, I'll stop here for a minute. Which of the drugs could enhance glucose uptake? All right, let's walk through these. A drug that activates the GLUT4 transporter. Well, you might recall that GLUT4 is the uniport transporter that brings glucose in to liver and muscle. And so, yeah, if we increase the activation of those transporters, that could enhance glucose uptake. So A seems to be a true answer. A drug that activates insulin receptors. Okay, a drug that assists in the activation or by itself activates insulin receptors is going to trigger the pathway that leads to glucose uptake. And so, yeah, B could be a correct answer because it would increase glucose uptake in liver and muscle. A drug that activates the MAP kinase pathway. Well, the MAP kinase pathway is downstream of insulin receptors. So if we have a drug that activates that pathway, then we could activate the pathway even without activating insulin receptors. And so that could also lead to an increased glucose uptake. A drug that slows tyrosine dephosphorylation. Slows dephosphorylation. So this is a double negative. It slows the dephosphorylation of tyrosines. So it's going to assist in tyrosine phosphorylation, which is the second step of activation of insulin receptors, binding, dimerization, and then cross phosphorylation of the receptor subunits on tyrosine residues. So if we slow tyrosine dephosphorylation, we allow the receptors to stay active longer. So I think the correct answer is all of the above. Yep. Now, let me just be clear that this drug, a drug that activates the MAP kinase pathway, is a terrible idea because activation of the MAP kinase pathway non-specifically could lead to all kinds of terrible effects, including cancer. So that there is no therapeutic target there that would lead this to be a good idea. But the other drugs, drugs that activate the transporter, drugs that activate the transporter, drugs that activate the receptors, drugs that, well, I got to say that this is probably a bad idea too, because it also will cause cancer. But the other ones, they're all possible. They all could enhance glucose uptake, but only 2 of them are good ideas. So now the remaining slides in this review presentation are really the slides from the section of the course that I think are most important. And I'm not going to reteach them. I'm not going to try to teach 4 or 5 hours of lecture in the next 30 or so minutes. I'm just not going to do it. But I think you should go through these slides and in your mind, or even on a piece of paper, narrate them. So what you might do is look at a slide like this, remind yourself of the anatomy, the hypothalamus, the median eminence in the infundibulum, the anterior pituitary and the posterior pituitary, and what makes them different. What hormones are released where? What hormones are released in the median eminence? Which hormones are released from the specific cell types that you should be able to name of the anterior pituitary, and which hormones are released by the posterior pituitary, and then what they do. So narrating the story of this slide is a good way to review the core concepts that are illustrated by this one slide. The next slide is pretty much the same thing in more of a flow chart way. And so this slide is a good reminder, but you might use your hand or a piece of paper to cover up the details. So you could, for example, cover up what's going on here and what's going on here and say, okay, what happens with TRH? TRH is released into the median eminence. It stimulates the thyrotrophs to release TSH, which goes through the systemic circulation to stimulate the thyroid gland to release T3 and T4. So by covering up and then revealing answers, you can narrate this story from memory and then reveal to check your answers. So that's how I would study this. I might take take the marker or just a piece of paper and block everything out. And then try to tell the various stories. And then reveal them to check my answer. Here we had a table like this in the very first class of the endocrine section. We have now discussed all of these hormones and all of these hormones. We have discussed all of the glands and that release the hormones, and we have talked about some of the most important effects. So again, I think you could use this slide now to review and re-solidify your knowledge about the hormones, the glands, and the effects of hormones. We did not talk about these, so let's make sure that none of these are going to be on the test. Well, that's not true. So we will, chorionic gonadotropin is a hormone that we talked about that you are responsible for knowing, that it comes from the placenta and that it is important for maintenance of the corpus luteum, the mammary gland, and the mammary development and maintenance of the mammary gland. We did talk about prostaglandins, which are a class of eicosanoids that are released from the uterus during stretch and are important in labor and delivery, among many other things. So we did talk about one example of eicosanoid. So the things that I've just highlighted on this slide, even though they are not, even though the placenta and the uterus are not typically considered sources of endocrine signals, they can be. And these two that I've highlighted here are topics that we did cover. The rest of them, GI tract, heart, kidney, we talk about in AMP2, and we haven't talked very much about the pineal at all. So pineal, release of melatonin, and circadian rhythms are not something that I would test you about in either course, actually. This slide reminds you of the process of the synthesis of peptide or protein hormones. So you should be able to narrate this slide. I'm going to resist the temptation to do it myself. You should be able to narrate this slide and describe how we get from messenger RNA to a precursor hormone in the lumen of the endoplasmic reticulum. And you should be thinking about what is a pre-pro hormone, what is a pro hormone, and what is a hormone. So this slide only describes pro-hormone being cut into hormones, but all of them come as a precursor, a pre-hormone or a pre-pro hormone. The cutting of the signal sequence is what generates, which goes from the pre form to either the pro or to the final hormone form, depending on the hormone. You should be able to narrate this story associated with what happens in the endoplasmic reticulum after a peptide hormone is synthesized by a ribosome docked on the surface of the endoplasmic reticulum. This is the story of peptide hormone synthesis and release. This slide was the example that shows that processing of a pro hormone, so pre-pro opiomelanocortin had a signal sequence that was cut off. Pro-opiomelanocortin is what is shown here. This is pro-opiomelanocortin after the signal sequence was cut off, and processing, cleavage of the pro-hormone releases ACTH and other hormones from this single precursor. I will not ask you for the details of what is ACTH versus MSH, but you should know that ACTH comes from a precursor, pro-opiomelanocortin, that also is the precursor for other hormones. I will not expect you to know the structures of the various steroid hormones, but I could ask you to identify a steroid hormone if I gave you a picture of one versus a picture of, say, insulin, and be able to say which was insulin and which was a steroid because of this four-ring structure that is unique or common to all of the steroid hormones because they all derive from cholesterol. I will not ask you to know the pathway for synthesis of the steroid hormones, but I think I could expect you to know that the estrogens are produced from the androgens. These here are the androgens. The androgens are the precursors to the estrogens. That's something that I think you could know. And I think you could know that the precursors to the androgens are also the precursors to cortisol and aldosterone. So that is a concept, and that is a concept that I think you should know. Do not memorize the details, but know the general flow. They all flow from cholesterol. The precursors, the hormones that are generated from cholesterol are the precursors to both the androgens and hence to the estrogens and to cortisol and to aldosterone. And you should know that if there is a defect in one of these pathways, so if there is a defect here, the precursor below, or on the far side of the defect, the precursor will tend to increase because its pathway for conversion has been inhibited. And so if there is an increase in the precursor, there could be increases of its precursor because of the equilibrium condition. And the increase of a precursor could lead to increased production of alternative pathways of metabolism. That concept of inhibition of an enzyme leading to stimulation upstream and stimulation of alternative pathways is a concept that I could challenge you with on the exam. A bunch of hormones. So this is going into the detail of the hormones from the hypothalamus. So we already sort of covered this when we were looking at the anatomy of the hypothalamus and the anterior pituitary. But you might want to be able to reproduce this table from memory. And this table also highlights the names of the cells, the cell types that are important in the anterior pituitary. Anterior pituitary hormones, their targets, and their effects. I quizzed you on this, but I could also expect you to know this for the exam. Posterior pituitary. We've talked about vasopressin and how it's important for the retention of water in the setting of either dehydration or excess hydration, and oxytocin and its important role in ejection of milk from the mammary gland, contraction of the uterus from during labor and childbirth, and its effects on behavior. We didn't talk about that in too much detail, but oxytocin has effects on pair bonding, mating, maternal and paternal instincts. By the way, oxytocin, there's a drug called Pitocin, which is actually just a synthetic version of oxytocin that is used to induce contraction of the uterus when a pregnancy needs to be induced. So you might have heard about somebody who had to have their labor induced because the pregnancy was going on too long. And the drug that is used to induce labor is Pitocin, which is really just oxytocin. The term I think that they use is a Pitocin drip. They use the Pitocin, oxytocin has to be administered intravenously. So in this case, the mother would have a IV line introduced, and Pitocin would be introduced at low levels until labor was initiated. We talked about the HPA axis, and so you should review the feed forward and feedback pathways for the HPA axis. You can look at it this way, you can look at it this way. All of these are the feed forward pathways, and know that cortisol and ACTH exert negative feedback on the hypothalamus, and that ACTH exerts negative feedback on the hypothalamus. So understanding the stress response is an important part of what we did. This is a cartoon that illustrates the stimulus and the response of pancreatic beta cells. You should know the mechanisms that are illustrated in this slide. You should be able to tell this story from memory, not just from looking at the slide. So looking at the slide should remind you, and then you could be able to tell the story of what stimulates insulin release by pancreatic beta cells from memory. This slide illustrates the story of how insulin interacting with insulin receptors triggers the storage of glucose in liver, fat, and muscle. This slide is not very informative because it includes this black box of signal transduction, but you should be able to reproduce from memory those steps: dimerization, cross-phosphorylation, recruitment of adapter proteins, activation of a kinase cascade, changes in gene expression, increased expression of the glucose transporter. This slide illustrates the mechanism for the synthesis of the thyroid hormones. So you should be able to remember that there is thyroglobulin, a protein that has tyrosine residues that are iodinated and then transferred from one to the other to make the two-ring structure of T3 and T4. Knowing the difference between T3 with 3 iodines and T4 with 4 iodides, and remembering that iodide is gotten into these cells by a sodium iodide transporter, that really a high affinity iodide transporter that helps bring iodide into the lumen of the follicle cells, the thyroid follicle cells, and then passive diffusion is what brings the iodide into the lumen or the colloid of the thyroid gland, where the iodide is attached to those tyrosine residues. Then there is endocytosis, lysis, and release of T3 and T4. Male reproductive anatomy. Know the names of these structures. More male reproductive anatomy. Know the names of these structures. The different cell types in the male gonad. The Leydig cells that respond to LH that synthesize testosterone, and FSH, which stimulates the Sertoli cells of the seminiferous tubules to assist in spermatogenesis. Testosterone and FSH both stimulate the Sertoli cells to assist in spermatogenesis. The names of the cell types in spermatogenesis: spermatogonia, primary spermatocytes, secondary spermatocytes, and spermatids. I will not ask you to know which of these are diploid and which of these are haploid, but we start with a diploid cell and we end with a haploid cell, so you probably could know that from, well, you could know that from CGI. The endocrinology of male reproductive biology: GnRH, LH, Leydig cells, testosterone. negative feedback. FSH, Sertoli cells, testosterone in a paracrine way, stimulating the Sertoli cells in addition to FSH stimulation. Spermatogenesis. Other responses to testosterone, including hair, muscle, bone, behavior. Female reproductive anatomy, knowing the names of these structures, the ones that were highlighted in the original PowerPoint presentation. More female reproductive anatomy. Oogenesis. The names, the oogonia develop into primary oocytes and they develop in the fetus of the woman. That the second step of meiosis occurs after fertilization is an important, interesting tidbit of information. That we generate one ovum, one egg per oogonia, not 4, as we do in the case of spermatogenesis, because we generate, and by we, of course, I mean you women, we generate one secondary cell and one polar body in each of these stages of meiosis. This is a key slide that illustrates the names of the follicular and luteal phase of the female reproductive cycle, the menstrual cycle, the levels of estrogen and progesterone, the levels of LH and FSH, the surge in LH and FSH that leads to ovulation. So the physical changes and the endocrine changes during the female menstrual cycle are things to be able to use, to know and to use in the exam setting. The development of the ovarian follicle, from primordial follicles to primary follicles to secondary and then mature follicles, the structure of the mature follicle with the oocyte, the granulosa, and the theca on the outside, the structure of the ovulated oocyte with its corona radiata surrounding it, and then the development of the corpus luteum, which in the case of no pregnancy degenerates, but in the case of pregnancy, implantation, chorionic gonadotropin, is maintained throughout the 9 months of pregnancy to produce progesterone and estrogens. Another cartoon of the developing follicle, knowing the names of these structures. The only one on this slide that I don't use is cumulus oophorus. You don't have to know that one. And we didn't talk about the nucleus so much. But all the other names on this slide are parts of the structure of the follicle, the ovarian follicle, that you do need to know the name of. This is one of the slides that illustrates the negative feedback during the early and mid follicular phase, the transition to positive feedback at the end of the follicular phase that triggers ovulation, and from there we are now in the luteal phase, and then the return to negative feedback during the luteal phase, due primarily to high levels of progesterone secreted by the corpus luteum. So this is this image here. This is the corpus luteum after ovulation. Again, being able to walk through the positive and negative feedback cycles, feedback loops in the female reproductive cycle. When there is negative feedback, when there is positive feedback, and what that does. This summary slide is important, and you should be able to walk through all of the steps that are going on. The development of the follicle and ovulation associated with the LH and FSH surge that came from high levels of estrogen in the setting of low levels of progesterone that also helped to develop the uterine endometrium. The return to negative feedback due to the progesterone produced by the corpus luteum. In the case of no pregnancy, the degeneration of the corpus luteum, the loss of the non-maintenance of the proliferated uterine endometrium, the loss of the uterine endometrium, and menstrual bleeding. In the case of a pregnancy, the fertilization events, chorionic gonadotropin sustaining the corpus luteum to maintain high levels of progesterone and high levels of estrogen, high levels of uterine endometrial development during and after implantation, and the development of the placenta. So being able to walk through the cycle, whether it results in a pregnancy, as illustrated here, or does not result in a pregnancy, as illustrated here, are both things that you should be able to do. The steps associated with fertilization, knowing that there is enzymatic reactions, the acrosomal enzymes of the sperm are important for getting through the corona radiata and then getting through the zona pellucida. The cortical, after fusion of a sperm, the cortical reaction, the reaction that releases enzymes from the oocyte and changes the properties of the zona pellucida so that it no longer can be penetrated by sperm, preventing polyspermy. The fusion of the sperm and the nucleus, the fusion of the sperm with the egg also triggers that second meiotic event, and then fusion of the genomes to create the zygote. The positive feedback loops that trigger labor and delivery. Oxytocin from the posterior pituitary, stimulating contraction, which stimulates stretch, which stimulates the production of oxytocin. Contraction and stretch also stimulates contraction and stretch due to prostaglandins. So positive feedback loops that lead to labor, the development of the breast during lactation. So estrogen during pregnancy develops the gland. Progesterone during pregnancy helps the gland make these well-developed secretory alveoli at the end of the ducts of the mammary gland, into which milk, a mixture of sugar and protein and water, is secreted and stored. Prolactin is kept under negative control by dopamine, but then during breastfeeding, inhibition of dopamine release, inhibition of inhibition, so that causes stimulation of milk production by these now well-developed secretory alveoli. So prolactin, the loss of negative control, the loss of dopamine's negative control leads to stimulation of milk production. And the positive feed-forward events, breastfeeding, stimulation, inhibition of dopamine, prolactin secretion by the lactotrophs of the anterior pituitary, prolactin stimulation of milk synthesis by the alveoli, secretory alveoli of the mammary gland, stimulating oxytocin release, oxytocin leading to contraction of myoepithelial cells and the actual ejection of that synthesized milk. So I think this is the last slide. I hope you have seen the most important concepts to review and learn. I've tried to give you hints about how I would study for this exam. I'm happy to answer questions by email and or on the Moodle forum. If there are other information that I want you all to know about the exam, I will post it to Moodle. So you should be looking at Moodle, both the forum and just the main page, to look for announcements about the exam. Feel free to contact me at any time. And I'll see you when someday.